Okay, this lab is the enzyme indispensable catalyst lab. Enzymes are biological catalysts. And remember from the kinetics labs in the book, catalysts increase the rate of a reaction. It makes the reaction go faster. They don't get used up, right? But just their mere presence makes it go faster. Well, in the body, in the cell, we have catalysts all over the place, and they're enzymes. Enzymes are actually a type of protein. So it's the same class of molecule that muscles are made of, all right? Proteins. Proteins are made of amino acids. So catalysts are a very special type of protein. Enzymes are a very special type of protein. They are the catalysts. You've got thousands of different biochemical reactions, metabolism, and biosynthesis and such, and DNA replication that are occurring in your cells, and they're all catalyzed by enzymes. Now, normal kinetics relies on things like concentration of the reactants, the temperature, the pH, the surface area, things like that, to affect the rate of that reaction. Adding a catalyst is another way we can affect it by its presence again makes it go faster. But how do things like concentration, temperature, and such affect the activity of the enzyme? That's what we're trying to get at here. Well, I mean, we know the enzyme will make the reaction go faster. Right? We know that. But now, what about the temperature on that enzyme, right? If we keep it at body temperature, for example, how fast does it go? What if we make it cold? Does it make it go slower? Does we make it hot? Does it make it go faster? Normal reactants would behave that way, but maybe not enzymes, because enzymes are very complicated protein machines, if you will, and the effect of such things on them might be a little surprising, actually. So without giving away the answer, we'll go into this lab. The reaction that we're looking at is always the same in this lab. We're taking starch and we're breaking it down. All right, so the hydrolysis, the breakdown of starch. Of course, starch you can find in potatoes. Right? It's a very long, large, complex carbohydrate. Starch will react with iodine. It's actually not even a reaction, but what it does is the iodine can intercalate and get woven in, if you will. None of those words are quite right, but it gets trapped in this huge starch molecule, these little iodines. And when you form that complex, you get a nice dark, kind of purplish, bluish color. Right? That means your starch is intact now. As you break up the starch, it releases the iodine, and you lose that color. You get just the normal color of iodine, which is kind of a yellowish, goldish sort of. Okay, and so the breaking down of the starch is what we're looking at. The enzyme that catalyzes that breakdown is called amylase. Amylase is the enzyme, it's actually in your saliva, is the very first step of metabolism, actually. We're breaking down any large starches that you might eat. Enzymes work pretty well. When we have the enzyme present, starch gets broken down very quickly. If you don't have any enzyme present, that reaction is incredibly slow. In fact, so slow that we wouldn't, in a couple hours here, we're not going to see any evidence of it at all. So it's kind of like an on-off switch. Either it's there and it's being broken down, or it's not there and we don't see it. So as we change the temperature or as we change the pH, what we're doing is we're affecting the enzyme, who's called, again, amylase, its ability to catalyze the breakdown of starch. Right? And we will see the evidence of it by the color. So part A, let's look at that. Part A is basically preparing the solutions that we'll need and preparing the water baths. So over here, our first one, we actually have an actual little water bath here. There's just distilled water in here, and we set it so that its temperature is right at body temperature. As you can see, I've got my probe in there. 37 degrees Celsius is body temperature. And so this water bath will mimic the conditions inside the human body. Next, we have our boiling water bath. Obviously, way hotter than inside the human body. Right? 97 is what we're recording there. We've got the probe down in that bath there, and so just shy of 100, and it's boiling. Now our third condition will be over here, an ice water bath. So very cold, and I don't have a temperature probe on this one, but it's going to be just above zero, like 0.5 degrees Celsius. It's ice water after all. Right? So three conditions, three different temperatures.
Most of this lab we're going to do in the 37-degree one to mimic the human body. But we are going to want to investigate these two, the hot and the cold one, as well, to see how that might affect the enzyme's ability to work. Okay, step 2. So we've got our bath set up. Step 2 says get the 1% starch solution. So we have that made up here. It's ready to go. 1%, I'm sorry, 1% starch. Okay. So again, this is what the reactant of the reaction. This is the complex carbohydrate that will be broken down. Okay. In here, it's not really breaking down yet because it's a very slow reaction. There's water in there, there's starch, but it doesn't break down because it's really, really, really slow. But if I add the catalyst to it, then it breaks down very quickly. All right, so we've got that. Step 3 says get the amylase solution. And here we go. Here's our solution of amylase. It's already been diluted to the correct level. All right, alpha amylase. So this is the same enzyme that's in saliva. In fact, you could do this reaction or do this lab without actually pre-purchasing amylase. You could use your own saliva, although that might be a little weird. But you could do it because that's where this enzyme comes from, and that's where it's naturally found in both you and me. All right, so we have our amylase solution, and then we have our iodine reagent over here. Step 5 says, or step 4 says, obtain about 1 milliliter of iodine and add 1 milliliter of water. So we want to dilute this, 1 water and 1 iodine. So let's just go ahead and do that right now. I've got a little beaker here that will keep our iodine solution in. We don't need a lot of this. Remember, the iodine is just the indicator to tell us whether we have starch present or not. Okay, and so this is just to indicate, and therefore we won't need a whole lot. So there's 1 milliliter of iodine, and you can see the natural color of iodine. Well, let me dilute it first and then I'll hold it up. Here's some distilled water, so get 1 milliliter of that. And we'll swirl it up. And so, here, I'll hold it in front of this white. You can see it's kind of an amber, brownish, yellowish kind of a color. Okay, that's how normal iodine is going to look in solution. All right, so we have all of our solutions present and ready to go. Now, when we add the indicator to the starch, right, and see if it's there or not, to see the activity of the enzyme, we'll get different colors. Right? Again, if there is no starch present, then you're going to get that color, or maybe a lighter version of it, so you get kind of a goldish color. That would indicate that the enzyme was working very well, right? That we broke down all that starch, there's no more left, and you'll get that kind of a color. So that would indicate a very high level of enzyme activity, and we'll call that a number 3. When you're taking your data, as we go through this lab then, that would represent a number 3, a gold color, high activity of the enzyme. Boy, that enzyme is really working. Okay? And then we kind of go down in levels from there, and in the manual you have this table, and it says gold, that's the highest level of activity, again, number 3. A step down from that, if you got like a light brown color, as it's describing, well, you had moderate activity. The enzyme wasn't working as much as before, but you know, it was still there. Bluish color means low activity. In other words, when this indicator gets combined with some starch, it forms that complex and you start getting that blue color forming, okay? So blue color means, well, very low activity, very low. And finally, at the very lowest one here, a dark blue-black color, that would indicate no activity of the enzyme. All that starch is still there, it wasn't broken down, that enzyme was not working, okay? Or it wasn't present or something like that. And so we'll see different temperatures, again, different pHs, how does it affect that enzyme? So got it? We're going from a dark blue to blue, light brown, and gold, meaning 0, 1, 2, and 3 on the level of enzyme activity. Okay, part B. We've got our baths at different temperatures ready. We've got our solutions ready. Part B will begin our investigation by looking at concentration. But now, not of the starch, concentration of the enzyme. So if you add a little bit of enzyme, if you add a little more, if you add a little more, how does that affect the rate? I mean, maybe it's such that, hey, just a little bit of enzyme is all it takes and you max it out immediately, or maybe it will increase as you add more enzyme. I don't know. We're going to find out. All right. So the effect of concentration of enzyme, how much enzyme is present. All right, so let's go through the procedure. Step 1 says place 3 milliliters of a starch solution, and here we go. Here's my starch solution into each of 4 separate test tubes. So here's my 4 test tubes, 3 milliliters in each one. Okay, so there's 1 milliliter. 2, 3. 1, 2. And again, we don't need to be super careful on the volumes here. We want to have reasonably about the same in each test tube so we can compare them. And that will be good enough, so I'm not taking too great of pains to get these just perfect each time. I'm going to compare them afterwards, and if one's a little low, I'll go ahead and top it off. As long as they're about the same amount in each one, that will be fine. You know, when you're talking about an enzyme catalyzed reaction like this, the reactant, the starch in this case, is also called, in biochemistry language, the substrate. The substrate is the molecule which binds to the enzyme. And in this case, starch is the thing that binds to that enzyme and makes it work. So let me see. There's my 4. Hey, I did pretty good actually. They're all about, just about the same, 3 milliliters in each one. And so I'm pretty happy with that. And we're going to leave it just like so. Okay, so my substrate, that is to say the reactant of the reaction, there it is, the same in every single test tube. All right. Step 2, we're going to place the 4 test tubes plus the amylase tube. Here's the amylase tube, which I guess we need a little bit of that amylase solution. Let's see, we'll get probably about 5 milliliters or so of that amylase solution. And so this is the enzyme again. Enzyme is amylase in this lab. You can think of that always being the same, so you just don't mix up which is which. Amylase is enzyme. So here's my enzyme solution, ready to go, and my substrate, the starch solutions. Place all of those at 37 degrees in the water bath and wait for 5 minutes. So we're just trying to get them to come up to temperature. And so let's come over here. Here's my 37-degree water bath. And I have a test tube rack right in there. Okay, I don't know if you want to zoom in on that. That will allow them to come up to 37 degrees. It won't take very long, actually. We'll give it a couple minutes. They suggest 5 minutes in the protocol, so we'll give them a couple minutes. And I think while we do that, let's go ahead and prepare the actual plate that we'll do the test in, which is step 3 in the protocol for part B. So you can bring the camera over here. Let's go ahead and do this next part. All right, step 3, it says, hey, while we're waiting for the temperature to come to equilibrium in that 37 degrees, prepare the well plate here with starch by placing 1 drop of the iodine. Remember, here's the iodine that we mixed up with the water before, right? 1 drop of that iodine in each of the wells. And we have 3 rows, 4 tests, and we got 4 test tubes, so this is going to be perfect. Here, here, and here. So this is just the indicator again. We don't need a lot of it. And so just 1 drop in the bottom of each well. And again, there's the natural color of the iodine solution. I actually got 2 drops, I think, in that last one. I might go back and redo that one. The drops are clearly not highly reproducible. They're about the same size in each one. Not bad, but I think this very last one I'm just going to wipe it out with paper towel and redo that one because I'm quite positive I got 2 drops in this top one right here. I mean, I want a reasonably the same amount in each well. And that one was notably higher. Okay. There we go. That looks better. Some of them are a little higher, some are a little lower, but that's, I think, okay. That kind of variation should work. All right, so our test plate is ready to go. All right. And we have our 4 test tubes that are being brought up to 37 degrees. And the idea is we're going to put different amounts of our amylase solution in each one of those test tubes. And then we'll check to see what kind of activity we have. So, reading the next one, it says, in test tube number 1, let's actually go back over to the water bath here. In test tube number 1, we're not going to put any enzyme at all. That's going to be our control test. In test tube number 2, we're going to put 2 drops. Test tube 3, we're going to put 4 drops. And test tube 4, we'll put 6 drops. So 0, 2, 4, 6. We're going to increase the amount of enzyme, and remember, here's my enzyme right there, in each one of those. And it's all at 37 degrees right now, so it's at body temperature. All right, so it says, go ahead and put the drops in, and then step 7, immediately transfer 4 drops of each one of these solutions to the test plate and observe the colors. All right, and so, you know what, I'm going to bring that test plate over here, I think. That might be a little easier. I won't have to run back and forth on the lab bench. I'm just going to push this back a little more, give myself some room here. I'll get the test plate and bring that over. Wipe this down. All right. And we should be ready to go then. Let's bring my droppers over. Okay, so for the amylase, again, I'm going to just go ahead and do this right here in the water bath. I'll move my temperature probe back a little bit so I have more room. And hopefully, can you see down in what I'm doing on the camera? Good. So again, 0 drops will go into this first one, 2, 4, and 6. So here's my enzyme, the amylase. I'll go ahead and draw some up into here. And nothing in the first tube. In the second tube, 2 drops. 1, 2, 4 drops here. 1, 2, 3, 4, and 6 drops here. 1, 2, 3, 4, 5, 6. All right. I'm going to just go ahead and leave that in there. All right, so we mix it together. I'm just kind of wiping my finger off. This is probably not the best lab technique around, but I think we'll get just fine results. Also, because I'm going from the lowest to the highest concentration, if there is any little bit remaining on my finger, I'm actually not adding it. All right, so now 4 drops in each one of these, and I'm going to go right across the top row. Here's the one with nothing here, no enzyme. 1, 2, 3, 4. Cool. So I'm going to do a drop each time here. The one with a little bit of enzyme, the 2 drops of the enzyme. 1, 2, 3, 4. I'm going to leave those in there. The one with the 4 drops. 1, 2, 3, 4. Now what's interesting about this is, wow, they're all the same so far. Here's the one with 6. 2, 3, 4, 5, 6. So they're all the same color at this point. And you know what, I just realized the procedure says, hey, after you do that in 5 minutes, test it again. So let's get my stopwatch going. Here we go, and I will put that up here. Of course, it probably took me about 30 seconds to do those drops. That's much better, thank you. I'm going to stand so we can see the timer. That probably took me about a half a minute when I added it, and then I put it down there. So 5 minutes, we'll do it at 4 minutes and 30 seconds. I think that'll be about the same. So while we're waiting for 4 minutes and 30 seconds to go along, why are they all the same at this point, and what does the color mean? You can see that all 4 test tubes have a nice dark color to them, right? That dark blue, blackish almost color. That simply means that there's lots of starch. The starch is present, it is not broken down. Remember, that iodine is an indicator for that starch, and it's kind of cool when you drop the clear liquid onto an orange drop and then all of a sudden it just turns dark blue like that. But that's chemistry, that's why I love chemistry. So that is evidence that starch is present. Now, what's happening over here in our test tubes? Well, it's sitting at 37 degrees. We put the enzyme in there. The starch is in there. Hopefully, it is catalyzing the breakdown of the starch. And so in 5 minutes, we test it again. There's not going to be as much starch in those test tubes. And we might start seeing a difference, therefore, in the color of these. We'll do the same thing at 10 minutes as well. Enzymes do take a little bit of time. Some of them can be very, very fast in the body. But within a couple minutes, that's probably pretty average for an enzyme to work on its substrate. And that's what we're doing right now. We're just waiting for about 4 minutes and 30 seconds, which actually be 5 minutes according to the procedure, that we'll try again and we'll see, do we have activity? Okay, here we're at 4 minutes and 30 seconds, which is our 5 minute mark. So we'll use the second row this time, and we'll do the same thing. We'll add 4 drops and see if there's a change. So first, tube 1, where we put no enzyme at all. 1, 2, 3, 4. Yep, that starch is still present, just as we kind of expected. Tube 2, where we put only 2 drops of the enzyme. 1, 2, 3, 4. Looks pretty dark. Tube 3, where we put 4 drops of the enzyme. 1, 2, 3, 4. It's not quite as dark, and I don't know if you can see that well on the camera, but it is a little bit different. Tube 4, where we put 6 drops of the enzyme. 1, see that color change? It's a little different, right? 2, 3, 4. So that's very interesting. Where we see that we put the most enzyme, it is causing that reaction to occur. It is causing that starch to break down. And we see the evidence of it by the color change. Okay, there we go. 9:30, so plus the initial 30 seconds. Those enzymes now have had 10 minutes to chew on that starch to try to catalyze the breakdown of it. So once again, we'll go and we'll test them tube by tube. Tube 1, where again there was no enzyme, our control. 2, 3, 4 drops. Yep, starch is still there as expected. Tube 2, where we just put 2 drops of the enzyme. Let's see what that one's looking like now. 1, 2, 3, 4. Still pretty dark. We had a little bit of enzyme in there, but not enough yet to see the difference in the starch. The third tube, this is the one where there was 4 drops of the enzyme. 1, 2, 3, 4. Now here we definitely see the evidence that the enzymes have been working on the starch. How about tube 6, where I keep saying that, tube 4, where we put 6 drops of the enzyme. 1, 2, 3, 4. Wow, very light. In fact, that's so light, that's that gold color that we're looking at. So there's no more starch left in that one, is what we're seeing there. It has all been broken down. So the amount of enzyme that's present, 0, 2, 4, 6 drops for each column, really makes a difference for 0, 5, and 10 minutes on ours. So make sure you get that copied down, the data. And again, you want to score each one of these wells as either a 0, 1, 2, or 3, depending on how much enzyme activity we see evidence of. Okay, the last part of part B, Benedict's reagent again tests for non-reducing simple sugars. Well, they don't have to be simple, I guess, but non-reducing sugars anyway. We'll put 3 milliliters of Benedict's reagent in each one, then I'll talk about what that means. Okay, and so that's about 1.5 milliliters. So I'll do 2 squirts in each test tube. 1, 2. Beautiful, nice little blue color, right? Light blue. 1, 2. 1, 2. 1, 2. And I'm going to go ahead just a little bit more of that first test tube. It's a little bit low. Yeah, these are a little low compared to it too. Just to get them about the same. All right. So they're all nice and light blue. Let's transfer them to our boiling water bath here, which is at 100 degrees or just below 100 degrees, and we'll let them sit for about 3 or 4 minutes. So we'll reset the timer and we'll say go. All right, so what is a non-reducing sugar? While those go here. A non-reducing sugar is a sugar that cannot participate in a redox reaction where it is acting as a reducing agent. Many carbohydrates, many sugars that is to say, can act as a reducing agent. They cause other things to be reduced in a redox sort of a fashion. Starch is not a reducing sugar. It's too big and complex. It just doesn't have those available. Whereas if you break up starch into glucose, which is the same thing in your blood, by the way, blood sugar is glucose. If you break it up, then the individual glucoses will be reducing sugars. And so this should hopefully correspond with our plate as far as is the starch being broken up. And the Benedict's reagent should show us that. While this continues to heat, I am going to go grab a tong or something so I can take these test tubes out. Oh, actually, Dr. Shin will do that for me. You already see some color changes happening, I think, in these test tubes, but we'll give it a full 3 minutes. We're only at 1 minute and 20 seconds so far. We'll give that a full 3 minutes for that Benedict's reagent to work, and then we'll take those test tubes out so we can see the color of them. Okay, so if I'm looking at these labels correctly, they're starting to come off the test tubes, but this is number 1. Test tube number 1. This one is number 2. Here's number 3. And number 4. Okay, and maybe if I hold up a white paper behind it, you can see the different colors representing whether we have non-reducing sugars. And again, starch is a non-reducing sugar, whereas the glucose down here are reducing sugars. And so you see the difference as to whether or not starch has been broken up by our amylase. Okay, part C. Here we're investigating the effect of temperature. All right, so in a normal reaction, of course, the hotter you make it, the faster the reaction goes. Every 10 degree you increase, the reaction rate more or less doubles. That's our rule of thumb. Is that true for enzymes? Here we have a reaction that does not happen, the starch breaking down, right, unless our enzyme is present. So we're going to try this at 3 different temperatures. 37 degrees, that'll be our first temperature. 100 degrees, that'll be our hottest temperature. And then number 3, we'll put in the ice bath, that'll be the cold. Right? And will we see that same effect as far as the activity of the enzyme? Well, that's what we're going to find out. So, in the procedure, step 1, 3 milliliters of our starch solution. So here's our substrate in each one of the test tubes. Okay, so there is 1, 2, 3.
Second test tube, same idea. 1, 2, 3. 1, 2, 3. And let me just make sure I got about the same in each test tube. Just line those up. And, yep, that looks pretty good. Pretty much the same. Which is what I'm really after here. I just want to be able to compare one test tube to the other, so if I have about the same amount in each test tube, then that'll be comparable. Whether I'm a little above or below 3 milliliters is not as important. Okay. It then says place 1 tube in the boiling water, place 1 in the 37, and 1 in the ice bath. All right, so the first one again at 37. Let me just bring the whole rack over to this side. Okay, so test tube number 1 at 37 degrees. Test tube number 2, boiling. Test tube number 3 in the ice bath. There we are. And so those are the 3 temperatures we are testing. Step 2, add 4 drops of the amylase solution to 3 other test tubes and place them, 1 in each bath. So we need 3 more test tubes that will do the amylase. Perfect. Thank you. 1, 2, 3. And we want our amylase solution about 4 milliliters. No, I'm sorry. 4 drops. 4 drops, and not a whole lot. 1, 2, 3, 4. 1, 2, 3, 4. 1, 2, 3, 4. 4 drops in each one. And we just want to bring those to the same temperature, I believe. Let me just double check that. And place 1 in each bath, right? So here we go. We'll take our enzymes and add them to the bath right next to our starch. So we're getting them all to be the same temperature. Okay. And the cold one right there. All right, as those come to temperature, let's go ahead and prepare our plate. Yeah, the procedure says to give them about 10 minutes or so to come to temperature. You probably don't need quite that long. So we can prepare our plate while we're waiting for that. Let's see, how much time do we have so far? Let me just reset my timer here. My phone. I mean, they've probably been in there 2 minutes so far. You know, again, it's just a matter of getting into temperature, so the time isn't critical. Let's go ahead and add our drop of iodine indicator. And we don't need the whole plate this time. We're just going to do 3 of them. So 1 drop. 2 drops, and 3 drops. Okay, they've been sitting in their water baths for at least 8.5 minutes here, plus that initial time, so that's 10 minutes. So what we're going to do is combine them together. And so at the 37 degree, here is our amylase enzyme, and again, here's the starch. We'll go ahead and we'll pour that in to combine it together. Mix it. Put it back in. We'll do the same thing for the one that's 100 degrees. It's a little hot, so I'm going to take it out and put it in a test rack. Oops, like so. And yeah, okay, I can pick it up. Pour that in to combine it. Okay, and turn that to its hot temperature. And then the same thing for what's in the cold. The enzyme, the starch,
Combine that together and return it to its temperature. All right, so we're going to reset the timer now for, I'll just set it to 0 and start it up again. So we're going to allow them to react for 10 minutes. You remember on the initial part and this plate, at 10 minutes we definitely saw activity, and so we're going to let these guys go for 10 minutes at their respective temperatures, and then we'll test them. Okay, so the enzymes have had 10 minutes and 30 seconds or so to react. Let's test them. Here it is, the 37-degree one. Now, put that in the first spot in the test rack. The cold one over here. That was number 3, and then the one at 100 degrees. There. Okay. Let's go over here to our test plate. And we'll, as usual, put 4 drops in 3 spots on our plate, and we'll see how well these enzymes did. So, tube 1, where it was at 37 degrees body temperature. 1, 2, 3, 4. Just like that. Not dark blue, so clearly that enzyme was doing something. Tube 2, which was at 100 degrees temperature. 1, 2, 3, 4. Dark blue. That enzyme did nothing. And at 0 degrees, 1, 2, 3, 4. Also dark blue. That's a very different result that you'd find in normal chemical kinetics. So enzymes respond differently to temperature than a normal chemical reaction would. Part D. Here we're investigating pH. Remember, 7 is neutral pH, body temperature or body pH is about 7.4. Less than 7 are acids, greater than 7 are bases. And so, how do enzymes like amylase respond to pH? Here we've created 4 different buffers. Remember, buffers are solutions that hold the pH, they resist change to pH. So we got a 2.0, acidic, way acidic, right? A 4, which is mildly acidic there, a 7, so basically at body, and then a pH of 10, which is basic, above body pH. How will amylase respond under these conditions? That's what we'll find out. So, step 1, we need 3 milliliters of each buffer into some test tubes. Let me joggle this around a little bit just so it's easier to grab. All right. pH 2 test tube, we'll put 3 milliliters of buffer in there. 1, 2, 3. pH 4 buffer into its test tube. 1, 2, 3. Okay, pH 7. 1, 2, 3. And finally pH 10. 1, 2, 3. All right, so we have 4 different pHs that we can do this reaction in. From the previous part, we saw that 37 worked fairly well for enzyme body temperature. So we're going to go ahead and take those and put them in our 37-degree water bath. And I'm going to see if I can just kind of balance them in the back here, since those test tubes are actually too big to fit in my little testicle. But yeah, that seems to work. It's not the prettiest, but it works. All right, also, we have our substrate, the starch solution, and we're supposed to put 3 milliliters into each one. I've done that ahead of time just to save a little time, so we're going to go ahead and put those also at 37 degrees. So those are identical at this point. Those are just the starch solutions. Finally, it says to add 4 drops of the amylase solution to each buffer. All right, those are already over here. That is the enzyme again. We're putting the same amount in each one. So we can do that right here while it's in here. So hold it up though just to make sure that it drops down into it. 1, 2, 3, 4. 1, 2, 3, 4. 1, 2, 3, 4. And 5 into 10. 1, 2, 3, 4. Okay. So it's all set to go. We just need to let it have a little time to come up to the right pH, right temperature. So we'll give it about 5 minutes or so. The procedure says about 5 minutes. So I suppose we should reset this. Okay, so our solutions are up to temperature. They've been sitting about 6 minutes. So one by one, we're going to combine them together. Remember, this is the buffer in here. This was the pH 2 buffer, and it has our enzyme, and this is just our starch solution. And so we'll go ahead and we'll pour that over. That. Maybe I'll give it a mix or two like here, and we'll put it back in at 37 degrees. And we'll just go ahead and do that for each of our tubes. Here's the pH 4. Add the starch. Give it a mix. Okay. pH 7. Okay. And pH 10. Okay, so we've got our amylase, our starch, at the different pHs. Let's go ahead and start our timer off. First, the protocol says to give them 15 minutes to react. While that starts, let's go over here and just quickly prepare our test plate as usual. We've got our indicator solution of iodine, and we have 4 different solutions that we'll have to test. So we just need to drop in our 4 different wells. So drop in each one. 1, 2, 3, 4. All right, that doesn't take very long, right? All right, so we're ready. We just wait for 15 minutes back over here on our 37-degree water bath. And so we'll cut the video here and we'll come back in 15 minutes. Okay, so our enzymes have been in the 37-degree bath for 15 minutes. Let's go ahead and take them out. pH 2, 4, pH 7, and pH 10. And we'll take them over to our plate and we'll test them out, dropping 4 drops in each one. Okay, pH 2. 1, 2, 3, 4 drops. pH 4. 1, 2, 3, 4. pH 7. 2, 3, 4, and pH 10. 1, 2, 3, 4. Okay. So at different pHs, clearly that enzyme behaves differently. We have plenty of starch at the pH 2 and pH 4, which are our acidic environments. Here's pH 7, which would be very close to what's in the body, and pH 10, which is actually way above what's in the body. But in both of those cases, that enzyme seemed to function just fine, but it did not work very well down at the lower pHs. Okay, part E, our last part of the enzyme experiment, we're looking at inhibitors. So inhibitors are other molecules, chemicals that inhibit an enzyme from working, that interferes with it or blocks it or somehow turns that enzyme off so it does not function. We're going to test a couple different things to see if any of these are inhibitors for our enzyme amylase. We have sodium chloride, so normal table salt. We have ethanol, which is the alcohol in beer and wine and such. And then we have some heavy metals. We have silver nitrate, so that's going to put silver in the solution. We have mercury chloride, and so mercury, the heavy metal. And we have lead nitrate, and so mercury, lead, and silver. See, many times heavy metals such as those will serve as inhibitors to enzymes, so let's find out. Step 1, we need to place 4 drops of the amylase solution and 10 drops of the sodium chloride solution in a test tube. So, over here, in each case, we're going to need 4 drops of the amylase, which is our enzyme, in every one of these. And so I'm just going to go ahead right down the row and add 4 drops of our enzyme. 1, 2, 3, 4. This one. 1, 2, 3, 4. And then for the heavy metals, 1, 2, 3, 4. 1, 2, 3, 4, and 1, 2, 3, 4. Okay. We have our enzyme in each tube. And now we're going to add the solution of possible inhibitors. And so, how much do they want us to add here? 10 drops of each one. Just reading ahead. Yep, 10 drops of everything. So, sodium chloride. 2, 3, 4, 5, 6, 7, 8, 9, 10. Ethanol. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Okay, our silver nitrate. 7, 8, 9, 10. Mercury chloride, actually. 1, 2, 3, 4, 5, 6, 7, 8, 10. And the possible inhibitor, lead, as in lead nitrate. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Okay, so we have our enzyme and our possible inhibitors in each test tube. Place 3 milliliters of 1% starch in a second test tube. So as usual, our starch, our substrate, our reactant, we need 3 milliliters in a separate test tube for each. So 1, 2, 3 milliliters there. That does not look like 3 milliliters. I think I may have done something wrong on that. I'm going to pour that out and try again. for waste. One, two. That looks a little better. The other one looked a little low. I don't think I probably had some air pockets in there. Didn't get what I was hoping for. One, two, three. One, two, three. One, two, three. And lastly, one, two, three. Okay. So we've got in the test tubes our reagent and our enzymes with possible inhibitors. Place them all in the water bath, 37 degrees body temperature, and give them about 5 minutes to come up to temperature. So I think I'm going to just combine it like that, make it a little easier to carry it over. All right. So really these starches are all the same. I'm just going to go ahead and line them up right along the back here. We have 5 test tubes of starch, and then sodium chloride, ethanol, the silver, the mercury, and the lead. Okay, and we'll give those 5 minutes to come to temperature. Okay, our solutions should be up to temperature at the 37 degrees. And so here's our enzyme with sodium chloride and of course our starch. We'll combine them together. And do a quick mix. Actually, I'm going to put it right back at 37, I think. Put it that way. Here's our ethanol and starch. Our silver nitrate with the starch. The mercury. And lead. All right, so now we have our 5 test tubes all at 37 degrees here, and the only difference between them is what possible inhibitor we have added. So we'll go ahead and stop this, reset it, and we'll give them 15 minutes for the enzyme to possibly work, and then we'll test it. Which means first we need to get our test plate ready with the iodine drops. And you've seen me do this several times. We have 5 test tubes, so we just need to get 5 spots ready. Alright, so I guess we'll do one here, one there, and then maybe 3 down here. So that'll be for our sodium chloride, our ethanol, and then the silver, and the mercury, and the lead. Okay, we'll see you back in 15 minutes. Okay, our test tubes and solutions have been sitting for at least 15 minutes. Let's go test them. So bring them out of the 37 bath. All right, let's go find out what worked. Okay, as usual, we'll put 4 drops of each one and the indicator. So first of all, the one with the sodium chloride, salt. 1, 2, 3, 4. Going with the ethanol. 1, 2, 3, 4. Silver. 1, 2, 3, 4. Mercury. 1, 2, 3, 4. And finally lead. 1, 2, 3, 4. Well, there you have it. Sodium chloride, ethanol. Might surprise you, actually, what inhibits and what doesn't, but those definitely look like they were working, right? Then we had the silver. Looks like there was a side reaction happening to the silver, but I certainly don't have that color. In fact, I got the dark blue color as well. So I would call that dark blue, and there's a side reaction creating this creamy thing as well. And then these are clearly very dark blue. Right? And so looks like the heavy metals are definitely inhibiting it. Sodium chloride did not. That doesn't surprise me too much. We have a lot of salt in our body as well. Ethanol, that's kind of interesting. Ethanol will often interact and denature proteins, but in this case, didn't seem to phase it one bit. We definitely had activity there. So possible inhibitors, there you go. It sounds like the heavy metals inhibit amylase.